let's move on to the actual work, experimenting with open tools. One of these tools is as ubiquitous as it is elusive, light. Or maybe not that elusive, because those who know how to tame that light can do extraordinary things in domains like optogenetics. By combining genetic engineering with optics, we might someday be able, for example, to map all the neural networks of the human brain. Our next guest was recruited to Université Laval to establish a viral vector platform just as optogenetics was emerging and promising to revolutionize science. The viral vectors developed through this open science initiative are distributed broadly, and testing results are shared with the community. Please welcome Marie-Ève Paquet.
Hi, I'd like to thank the organizers for giving me the opportunity to share what we've been doing with the Canadian Neurophotonics Platform. And so let's start by saying that this initiative is the work of many people and many organizations who share the common goal of advancing and promoting the use of light and gene transfer technologies in neuroscience. So let's start with going back in time a little bit in 2008, which is when I joined the Centre de Recherche Cerveau. So at that time, the Neurophotonics Center at Cerveau was well established and under the leadership of Yves de Coninck and Paul de Coninck and a few others later on. And through that center, collaborations between physicists and neuroscientists were already emerging as very promising. Optogenetics was a few years old, but its transformative potential was already being recognized, and viral vectors were emerging as a key method for introducing these optogenetics tools in cells, but also in animals. Sorry. So at that time, the Quebec Pain Research Network, which is a FRQS network, decided to fund a viral vector and molecular biology platform to provide access to these optogenetics tools to the pain research community, but also to the larger neuroscience community. So the initial model that we had with the QPRN was to use the expertise of my team in molecular biology to customize the optogenetics tools and then to package them in viral vectors. So basically, we were working as in large collaborations between users and my team. And I think that this model worked really well and had an impact on the democratization of optogenetics tools, because it became much easier for groups to have access to the tools that they really needed without having to develop, the expertise in molecular biology in their own lab. So in the years after we started with this platform and with the QPRN funding and model, challenges were starting to emerge within the platform that were linked to both the use of the optogenetics tools themselves and their design, as well as the delivery vehicles. So in terms of the optogenetics, there were issues with intracellular targeting, robustness, levels of expression that were required to be quite high, and there was a discrepancy between the efficiency of the tools in vitro and in vivo. So people were making or designing tools and demonstrating their usability or in vitro, but then when users wanted to use them in live animals, they weren't working as efficiently. And in terms of the delivery, it became quite apparent that there was a need for a higher specificity in terms of targeting. There were differences, in the efficiency of viral vectors between different species and strains of mice. And the fact that viral vectors had to be injected with involving invasive surgeries was also a bit of a concern. So to further accelerate the development of this field, we came up with an expanded and more integrated model that was involving 4 cores with complementary expertise. So a protein engineering core, viral vectors, software people, and people developing hardware that were working together with testing labs and early adopters in a feedback loop. And this model emerged around 2016 and became reality thanks to some Brain Canada funding. And with that model, you know, our level of distribution, dissemination became much more significant. We introduced collaborations between, you know, new fields and new labs, and we also introduced the optimization part of the tools with that model. And data sharing also became part of the model, mostly within the labs, within the groups involved in the model, but still we were openly sharing data, and the communication was really important. So this new model was quite successful, especially for my viral vector group, but at the same time, the success brought some challenges that we needed to address in order to feed the model properly. And viral vectors had to adapt to the field of optogenetics and the need of the model. More significantly was the speed at which the field of optogenetics was moving. Basically, you know, new optogenetics tools were coming out on a regular basis. It was really hard to keep up. New AAV serotypes were coming out also. People were experimenting with modes of injection. So we had to basically work to improve the speed at which we were working to really keep up. The other thing that we had to work on was developing viral preps that were adapted to the reality of testing, since we were engaged in that optimization loop. So initially we were making viral vectors in the size of like 500 microliters, when you actually need nanoliters or microliters per injection. And so we ended up, you know, wasting quite a bit of material. So we worked hard to develop these protocols that were allowing to package quickly in a smaller volume, but with the same level of purity. We also had to basically focus on one type of vector because it was the most promising. So those were the AAVs. And the other thing we did was to work on quality control steps for our viral vectors. So basically, for the loop to work, for this optimization loop to work, we had to make sure that the viral vector quality was not part of the optimization steps. So we had to make sure that the quality was a constant, basically, and that we weren't having to optimize that part. We could optimize the configuration of the vector, you know, the serotype and all these things, but not the actual quality of the vector itself. So now we have a viral vector facility with 8 people working full-time in molecular biology, AAV production. We do a lot of research and development also. We've been packaging more than 300 vectors per year for the last few years. We have a wide range of size of viral preps. And our titers are quite high now and stable. And so even though also we're heavily involved in this optimization loop, a large part of the work that we're doing is also distributing the optimized tools to the community and making them available, you know, to a larger community. So we distribute viral vectors in Canada a lot, but also abroad in the US, in Europe, in Australia. And we have a large database of constructs that we can package in viral vectors. So you can see what we have on our websites, neurophotonics, tools.neurophotonics.ca, sorry. And we have a lot of things there. So the initial CNP funding from Brain Canada came to an end, and we decided to refine the model based on what have been the most successful parts, and also considering where the field of optogenetics was going. So we came up with the optogenetics and vectorology foundry, which is based on a design, build, test loop. So the design involves protein engineers like our colleague Robert Campbell at the University of Alberta. Building is basically building these tools into a viral vector for delivery, and then sending them to a number of testing labs who are able to test the functionality and efficiency of both the optogenetic tool and the delivery vehicle in various experimental paradigms and species. And so, of course, this involves, again, numerous labs, numerous institutions with very many experts that range from, protein engineering to rodents to lower vertebrates, human cells. So again, a large collaboration between people with quite a large set of expertise. So I'm going to give you a few examples of how the CNP has been successful at improving the optimization of tools. So here's an example of a near-infrared fluorescent calcium indicator that was developed by Robert Campbell at U of A, and now he's in Tokyo also. So he came up with the first version of the sensor, sent it to us. We packaged it, for being used in various experimental paradigms, different promoters, different serotypes. And then we sent it to a number of testing labs around the world who used their expertise and generated several data that was then sent back to Robert Campbell, and he used that to generate a second version of the sensor. So we went around this loop until there was a feeling that there was robust enough for a dissemination at a larger scale within the community. And then that was published also in Nature Methods. So we're working now in a similar fashion on a new sensor, a lactate sensor that's also being developed by Robert. So now he generated iLACO 1.1. We've packaged it in several configuration of promoters and serotypes. We've sent it to 5 or 6 labs, and then some data is already coming out, and with respect to which one is working best in which cell type, and so on. And so now this data is being transferred to Robert, and he's coming up with iLACO 2, which will then package again, considering the information and the data that was provided by the testing labs. And we'll do this loop again until we're up to a robust tool, which I've called iLACO X. And that tool is then going to be distributed to a larger community and is going to be available for people to use in their own experiment. And we'll be confident that this is a workable tool. So another example of a slightly different but very useful outcome of the CNP has been the result of work that we've done with Tim Murphy and Mathieu Vanny when he was in Tim's lab. So as I mentioned earlier, the fact that AAVs have had to be surgically injected has been a concern for several years. But to overcome that, Viviana Gradinaru at Caltech has engineered new serotypes of AAV, which can be injected intravenously and can make it across the BBB to transduce cells in the brain. So we obtained this capsid from Viviana and packaged the calcium sensor GCAMP in that serotype, sent it to Tim Murphy, and he's used it in his model of transcranial window, and he was able to show that not only the serotype could transduce a large number of cells across the brain, but he was also able to show that the levels of expression were high enough and even higher than what you could see with HR models that are much longer to generate. So basically now we have a very efficient, non-invasive mean of assessing functional signals from genetically encoded tools in vivo. And we expect that this strategy is going to be very useful in the future for testing other tools. So like I said in the couple previous slides, we were obtaining capsids from Viviana's lab. And so we've actually been working with numerous developers around the world who work in different aspects of optogenetics challenges. So people working on promoters, sensors, different AAVs. So we work with these guys and they enter our loop, and we help each other in a way that we, with the viral vector platform, we help disseminating the tools and them available to the larger community, which really, as a tool developer, this is something that you want. You want people to use your tool, and then they became broadly available. And they can also send us tools that are in development, and then they may benefit from our testing capabilities. So it's a win-win situation, and we've been working with me in that respect. So with this new DBT loop, we have an increasingly open resource. So basically, not only the viral vector tools and the optogenetics tools, but we're also aiming at sharing a lot of the data. For example, the primate data are going to be entered into this primate optogenetics open database. We're also, since the field of optogenetics we feel like it's really moving towards human applications. We're now working with the human tissue platform led by Reza Sharif. We're working with the EDDU at the Neuro for testing these tools in human iPS neurons derived from iPS cells. And we're still very interested in lower vertebrates, such as the zebrafish. And we're hoping to, again, engage with the zebrafish community, with Ed Ruthazer, Paul de Coninck, and get together with these guys and share a lot of the resources and use our loop to do that. So in terms of getting involved and sharing, if you're interested, we're going to initiate a series of seminars soon, probably early December, which we're going to call the Global Resource Seminar Series. And the idea here is going to be to start discussing, large initiatives that could lead to the generation of shared resources. So we're going to have Reza Sharif and Jean Ouellet on human tissue accessibility and experimental approaches for using human tissue. Keith Marai and Sébastien Tremblay from the Pratt Lab at UPenn are going to talk about primate optogenetics. Isabelle Aubert from Sunnybrook in Toronto. So, keep an eye open for that. We also have the Canadian Neurophotonics Platform satellite meeting at Cannes. And of course, our website, you can visit anytime or be in touch with me. So I just want to finish by saying that this, of course, is a team effort. So here are the names of all the PIs that we've worked with. And I just want to say that, of course, these are the names of the PIs, but very importantly, the people in these guys' lab have been really instrumental. And our model also involves funding, research professionals to do these types of testing. So I want to thank them, even though I can't name them all. I would like to thank also Mario Méthode for coordinating this effort over the years, and of course my own people in my lab who are making all these tools possible. Thank you.